So the rest of our labs, we have like general sensation, vision, hearing and gustation and olfaction. Lab review, although lab review is probably going to include some finishing up some of the labs, although hopefully not. But basically the next 3 labs are all about senses. Today's lab is the general senses. The general senses, these are the ones that are basically all over your body. Includes, touch, pressure, temperature, pain. They're all over. They tend to use simple receptors. Most of the time, the receptors are simple little dendrites or modified dendrites off of a neuron or something. Versus the other stuff we're going to be talking about next week, which are going to be the special senses. So versus. These are in the head, complex receptors. This is like vision and hearing and equilibrium. And olfaction, which is the fancy word for smell. And gustation, which is the fancy word for taste. So we'll be covering these next week. Today, we are going to just focus here on some of the basic ideas around the senses in general, and then we'll talk about the general senses. So receptors are the things that are going to be at the core of your senses. And receptors are doing what we call sensory transduction. So this word, it's important you understand this concept because it's going to be at the core of all of the senses. And the basic idea here is the stimulus that you are detecting with your senses, with your receptors. There are different kinds of energies in the world. It might be light, or it might be some physical pressure, or it might be the presence or absence of some molecules that should be part of taste. Or it might be changes in temperature. So what you're actually responding to or noticing, they're all different flavors, but the only thing your brain understands, your nervous system works on voltage changes across the membrane, right? That's what a neuron knows, is like, oh, things are depolarizing or repolarizing. The brain only understands electrical signals. So if there is light coming in and your brain wants to know there's light, there's got to be some receptor that has the ability to convert light into some electrical signal, which then the brain will understand. If you want to understand touch, somebody's touching your nose, there has to be some receptor that can somehow convert that physical deformation of your skin into some electrical signal, which goes to your brain. So your brain says, oh, there's somebody touching my nose. If you want to taste sweet, you've got to have some receptor on your tongue that actually can detect a sugar molecule, and detecting a sugar molecule creates an electrical signal, sends to your brain. So your brain says, oh, there's sweet in my mouth. So sensory transduction is basically, translate the stimulus, whatever it is, into an electrical signal for the nervous system to understand. So this is going to be a recurring theme. We're going to talk about kind of how this works. We'll spend a lot of time looking at the eye and how does the eye, and specifically the rods and cones, create electrical signals in response to light. Other things like the taste buds are going to be really easy, right? Taste buds. How could you detect sugar and turn it into an electrical signal? How could you do it? If you wanted to make like the backbone of a taste bud that detects sweet, how would you build something that would create an electrical signal upon exposure to simple sugars?
Have a receptor for sugar and then have it depolarize the membrane.
Yeah, if you just had a receptor for sugar, and when that receptor binds sugar, it opens up a sodium channel, bing, you're done, right? There you've got the presence of a sugar molecule created an electrical change on the membrane, right? So sometimes the transduction can be relatively straightforward. Sometimes it can be much more complicated, as like when we get to the eyes, for instance. So, but make sure you just understand in general, that's what all of these receptors are doing, whether they're photoreceptors or mechanoreceptors or temperature receptors. They somehow convert the stimulus into an electrical signal. Another thing we should talk about is the idea of adaptation. So adaptation, so right now you are sitting in front of your computer watching me talk on Zoom, and you're probably not thinking about all the cloth on your skin. When you first pulled your clothes on this morning, you probably felt it rubbing against your skin or some pressure or this or that. And then after a while, you kind of don't feel it anymore. Or you put your glasses on your face and you definitely felt them land and rest on your nose and on your ears, and you haven't thought about that since. Or the temperature, assuming your temperature in your house is okay, maybe when you walked into the room, it was a little hotter or colder from the room you walked from. But then as long as it's in a reasonable range, you're sitting there, you don't even really notice the temperature, and the temperature just kind of goes away. All of that is this idea of adaptation. This is basically where you stop responding to a constant stimulus. Right? I see some people with ponytails. When you first pull your hair back for your ponytail, you feel your hair pulling back. But assuming you're not doing it so tightly that it's in pain the whole time, eventually you kind of forget you got a ponytail. Um, so adaptation happens on many different levels. We'll get into some of the weirder examples of it later, like in the visual system, we can get some really cool illusions going on and stuff due to visual adaptation. Today's lab looks more just at touch receptor adaptation, right? The fact that you're constantly not aware of all the clothes pushing against your skin, although you have receptors that, that are detecting touch and things like that. So what's the advantage of the fact that almost all your senses adapt?
You're not getting constantly inundated, so you can pay
Yeah,
attention to important stuff.
exactly, right? Everyone heard Dana there? It's like you're not being, you are being constantly inundated, but at least it takes it out of your conscious perception. So then you are able to pay attention to things that are more important. In general, if things change, like if there is some change, the receptor wakes up. But then if everything's cool and then it's stable in this new place, it'll go. So it's like if there's something changes, your nervous system is like, oh, let me check it out. And then if it's nothing that's going to be dangerous or edible or whatever, you just kind of like go back into putting your attention into something else that might be more important. So just important concept. Okay, we'll see it in a variety of senses. But today we're mainly looking at the idea of touch receptors and actually temperature receptors we're going to look at today as well in terms of their adaptation. And we'll talk about that in more detail in a little bit. So adaptation is an important concept. Thresholds. We don't have to talk about because you know what that is, like the minimum that you can just tell. The thresholds that we're going to be doing in our lab today is going to be like 2-point discrimination. Two-point discrimination is if here's my finger, but that's a bad finger. And I take 2 points. Here's some distance X. What's the minimum distance where they can be apart that I still feel it's 2, right? At some point, it could be if they're this close, it's like I only feel it 1. Whereas if they're this far apart from each other, I can definitely tell it's 2 different needles on my finger. And somewhere there's some minimum distance where you put the needles just close enough that it still feels like 2, but any closer, you just feel like 1. That's what we're going to be measuring as part of our lab today, is like, what's the closest you can have those 2 needles together and still know that it's 2 needles instead of 1? What is going to determine that distance? How are you going to be able, what's going to determine how close you can put these pins and still notice it's 2 instead of 1?
Would it be the receptors in your finger? Like, are you on a different one?
Yeah, exactly. So it's going to have to do with the receptors, right? If, let's say I only had 1 big receptor in my finger, there's no way it could ever tell 1 versus 2 because it just knows there's pushing or there's no pushing. It can't really tell any discrimination beyond that. Let's say I had 3 receptors on my finger here, like this. If this was my finger, I could definitely tell the difference between 2 yellow ones because my brain is saying yes, area with no, another area with yes. All right, there's got to be 2 points on my finger because that's the only solution that solves for positive receptor, negative receptor, positive receptor. This receptor configuration, though, could not tell the difference between 2 green ones right next to each other versus just 1 big one right there, right? So for this person, they could tell the difference that the 2 yellow ones were separate, but they couldn't tell that the green ones were separate. What if I wanted to have a situation where I could even tell the green ones were separate? How would I have to set up my finger? What would the condition have to be?
Do you mean like two small receptors, like right below the green one? Both
right?
the
Or
green
just a really high density of receptors, right? So there's such a high density of receptors that even when they're this close, they're still on, off, on. So I can tell that this is two and not one. At some point, even these will get close enough where I'm going to get too close for this particular density of touch receptors under my skin. And then it'll only feel like one instead of two, right? So the 2-point discrimination is going to depend totally on how dense the touch receptors are under your skin. Again, these touch receptors, these are the things doing the sensory transduction, translating mechanical stretch of your skin to an electrical signal, right? And so part of the lab is just doing this on different parts of the body. Do it on your fingers, do it on your lips, do it on your face, do it on your arm, do it on the back of your neck, do it on the back of your calf. Just from the results of our nerve stimulation lab, what might you expect about comparing 2-point discrimination on the fingers versus on the back of the calf, and why?
Threshold would be lower on the fingers because there's more sensory receptors than on the calf.
Yeah, okay, so the same kind of thing. You're looking at a different sense, but if you are, from our data from the nerve stimulation lab, we got some pretty good evidence that there's higher densities of receptors on the fingers to trigger off with the electrical stimulus that we were using for that lab. So that's going to mean if we have a higher density of little receptors that we're probably going to have better 2-point discrimination as well compared to the calf. If you have less density of receptors, it's going to be more like that situation I was showing you where there's not as many receptors, so you have much less ability to discriminate if things are really close. Right, so this is going to kind of tie in to what we found in the nerve stimulation lab. Another thing we're going to do that's related to this, that is a little different, but related, and is going to also depend on the density of receptors under your skin, is going to be this task called touch localization. So this, I don't know everybody's like kind of shelter in place situation. Hopefully most of you have other humans that you interact with. If you do, you definitely want to try this. Do this to other people, have them do it to you. Especially if you've never done this, it's really counterintuitive and kind of weird what comes out of it. The basic idea with touch localization is like, you'll have their hand. And you take a pen and you make a dot there. And you really push it on there. You're not trying to trick them or fool them or anything. You basically make a dot, really kind of make sure it's really, you feel it. And then you take this away, and then they then come in with their other hand and a pen, and they try to find what is the exact spot you were touching. And again, it's not like a trick. You don't have to just go in. It's like, oh, you made your spot. It's like, you can feel around. It's like, yeah, I really think it's right there. And they put a dot exactly where they think you were been touching them. And then you can measure what is the distance, the difference between the actual place that they were touched and the place when they tried their very best to reproduce the place they were touched, right? And obviously, this is also going to depend on your receptor array under the skin. The information that your brain gets about where that first touch was is limited by how accurate and how dense the receptor array is underneath, as well as your ability to feel and localize, the dot that you're putting there. So if you've never done this, I think you will be like kind of surprised at how loosey-goosey your perception of reality actually is. Like you can, of course, and that sounds stupid. Of course you can find it and touch it and get it again. I think you'll be, if you haven't done it, you'll be surprised at how bad we can be at this kind of thing. Right, so this is going to be another part of the lab, touch localization. It's related to the 2-point discrimination in that it's going to actually be limited. Your performance is going to be limited by how dense the touch receptors are under the skin, but it's a slightly different way of looking at it. But you're going to expect kind of a similar, a correspondence. If you're better at 2-point discrimination at a certain part of your body, you're probably going to be better at touch localization on that part of the body as well, because you just have denser receptors, more of an ability to actually, zero in where exactly did that happen on my skin. So let's, I'm going to share screen. Let's see. All right, so this is just kind of showing, this is a picture of the skin, and these Meissner corpuscles are, and the Merkel receptors are both things that detect touch, actually can detect kind of vibration, Meissner's. So this, when I'm talking about the little receptors under your skin that are converting the stretch or bending of your skin into electrical signals, it's these little things, these like Merkel cells and the Meissner corpuscles, and then send messages down their axons to your somatosensory cortex, the postcentral gyrus. So your brain has information that you're being touched. Some of these other ones, these free nerve endings, these are what temperature receptors look like or pain receptors look like. They don't even look like much of anything until you actually expose them to different stimuli. You don't even know what kind of receptor it is. You can't tell from looking at it. But so Pacinian corpuscles, these are like kind of deeper pressure, things like that. Right, so for 2-point discrimination, you're just going to have like calipers. Basically, if you're doing this at home, you can use like a compass. You can try to make something up like with a toothpick. Yeah, it could be toothpicks, or probably what makes sense. If you have like a stiff paper clip like this, right? You could like bend it until they're really close and see. And when you're doing this, you really aren't trying to fool them. If you're doing it on their face, really put it there. Is that 1 or 2? You're not going like, boom, is that 1 or 2? You really want to check out how good is their sensory, discrimination, not when you're trying to trick it, but when you're really giving them a chance. Is that 1 or is that 2 on your skin? And then they have to say, and if they feel that as only 1, Make them a little wider. Is that 1 or 2? Find out what is the distance that these are where they definitively know it's 2 and not 1. You know, and then you can just get out a ruler and you can measure what is that distance between the 2 points when they could really tell it's 2 instead of 1. If they feel it as 2, double check. Hit them with just a single thing and make sure they feel it as 1, particularly on the face or things like that. It's sometimes, a lot of times you have people seem like they're insanely good at this task, and then you hit them with just a single point on their skin and they still are convinced they're feeling 2. So it's kind of similar to what we were talking about with the nerve stimulation lab and the false positives. This is another case. You really want to double check. Don't just assume. If they say, oh, I feel, you know, these points at like 2 centimeters apart on the back of my calf, nobody does. It's usually like an inch apart before they can feel it as 2 on the back of the calf. So if they start feeling stuff really close, double check and make sure that they're not just noticing something that isn't there. So, you know, we have data for this. So if you don't have somebody to do this on at home, you know, you can just use the data and write it up. But I do recommend you do it with somebody if you have access to somebody who's willing to play along, because it's pretty cool. Tactile localization, again, this is what I was just talking about. You take one colored pen, and again, you are going to do this in a way that is not fooling them. It's really giving them a chance. You know, if I have, right, again, my hand, I'll take one pen. This is like a blue pen. Really, really make sure they really feel it. You're not just going boom, boom. Give them a chance. There's, oh, make sure there's a color there. Okay, there's a color, there's a little blue dot. And then I'm coming in, my eyes are closed, and I'm just feeling, where was that? And I think it's there. In fact, yeah, I mean, even when I just did it on myself, you know, they're close, but they're not on top of each other, right? So you're going to do something like that. You do it a few times at each of the places that we talk about in the lab manual. It's like on the palm, on the fingertip, on the ventral forearm means the front, ventral's belly side, anatomical position. You remember your anatomy, anatomical position, palms forward, standing up, ventral forearm is like right here. Adaptation of touch receptors. This is basically going to be putting coins on top of your arm, and you'll feel it when you put them there. And after a little while, you won't feel them at all, which is again kind of trippy because they're there. And no matter how much you try to reach for it, it's like you can't tell that they're there or not. You know, so partly you do this just to really believe this exists, and second, to kind of quantify, like, how long does it take? When you are doing this, if you're doing this at home, you want to kind of insulate the little coins. If the coins are cold, if they're metal, you might still feel just the temperature getting conducted away from your skin, which is different than the touch receptors adapting. So, or if you have like a little wooden, if you had a wooden nickel or something, so either wrap the coin in paper so it doesn't conduct heat, or use something that doesn't conduct, it has some weight so you can feel it, but isn't metal. which is going to continue to wick heat away, which you are going to feel. So again, when you're doing these, we have videos that you can look at, but I do recommend doing it at home as well, because that's where you're really gonna have fun with it and really get a better sense that it's real. So let's take a little break, and then we'll talk about the other part, the temperature and the pain. Okay, welcome back. So another part of the general senses is temperature. And, temperature, I'm not going to get into the detail, even though we talk about cold receptors and heat receptors. The more people have looked into what this temperature really, it's a little more complicated, and very much also temperature receptors adapt. They tend to respond to changes in temperature, but not necessarily responding if the temperature, again, is in a reasonable range and it's just staying constant. At that point, temperature receptors tend to adapt, meaning they stop responding. And so we have a couple of exercises here we call part 4. We can share screen. This part 4, adaptation of temperature receptors. This is something I would like you to do at home. This, you don't even need another person. So I can just say, for sure, do this. The things that require another person for the touch stuff, if you don't have someone to do it with, just, use the group data and, record what you find. If you do have somebody, I really recommend you do actually do it and put your observations as well as the group data as part of your write-up. For temperature, you are going to do at home, and I would like to spend a little bit of time being more explicit about how you do it, because I know where people tend to get a little confused on this. So adaptation of temperature receptors. So, it says, a finger bowl is just a bowl. It could be a cereal bowl. It doesn't have to, it's just a bowl big enough to like put your hand in. So you've got a bowl, and for the first part, you put in water. So this is like part 1. It says, around 43 degrees C. This is, warm, but not pain. Like when you put your hand in there, even if you don't have an actual thermometer to check it out, you should put your hand in there and it's like, oh, that's warm. But it shouldn't be. If you put your hand in there and after a few moments, you're like, oh, that's warm. Oh, that's getting uncomfortable. That's too hot. It shouldn't be about pain. We're going to talk about pain a little later. Pain is something different. And all your receptors, all your senses turn into pain when they get far enough to an extreme of their range, right? Too much touch or pressure starts becoming, oh, stop that. That's painful. Or that's hot. That's really too hot. That's cold. That's really too cold. That's loud. That's really too loud. So pain is not what we're going for here. We're just going for warm, nice and warm. And we are thinking about temperature. So when you are describing your observations, observe your perception of temperature. Like, does my hand feel warm or cooler? Or we're going to compare how the right and the left hand feel, in terms of temperature. So you're going to like put a hand here in this warm water and describe, write down, just, you have to use your other hand. So use your non-dominant hand to put it in the water. And then you can use your other hand, or if you have your study buddy at home, you can have them record the data as you talk to them. You describe, what does it feel like when you first put your hand in the water? Hopefully it's like, it feels warmer than the other hand now, because you're putting it into warm water. And then you're going to leave it in for 1 minute. After 1 minute, you are going to describe, how does that hand feel compared to how it originally felt when you first put it in. So, and again, in terms of temperature, not, oh, it's feeling tingly. Tingly is not temperature. That's where I think I want to be clear. You really want to think about the temperature experience. And then after a minute, you're going to put your other hand in the water as well. And then you can, so now you have this hand that's been in there already for 1 minute, and this hand that just got dunked. And you want to compare, what is the experience in the hand that's already been in there for a minute versus a hand that just freshly got dunked? And describe in terms of temperature, this hand feels warmer or cooler or something, or the same, it depends what it is. So, follow the procedure in the manual, but make sure you are observing, what does the temperature feel like in one hand versus the other hand? And again, make sure that the water is warm. If either hand, if you start your observation is, well, my hand is in pain, it's like, no, stop, take your hand out, use cooler water. It shouldn't be in pain. So that is like this part 1, is just use 1 bowl of warm water. And again, from adaptation, what would be your expectation if this hand goes into the warm water and has been there for a while? What do you think is going to happen to the experience of temperature?
It wouldn't be as warm after a minute, or it wouldn't feel as warm.
Yeah, it's probably not going to feel. As soon as you put it in this thing that's warmer than it's been, you're going to notice, oh, this is warm. But as that becomes the new normal, you're going to stop responding to that. So, and as you put your second hand, which is now fresh in the warm water, you're probably going to notice interesting things. So again, compare in terms of the temperature. So that's part 1. Part 2. This one I really like. This, I mean, I did this actually when I was a kid even. I remember reading about this and doing it when I was, I think I was like in junior high the first time I did this. And it was like the coolest thing ever. It was like part of what kind of started getting me into science and stuff. So for part 2, you're going to need 3 bowls. And they all have water. This takes a little bit of setup. And here you need to have 3 temperatures. This is going to be cool, but not too cold or painful. Right? This is not ice water. This is just kind of cool water. You're going to have the warm water, but not painful. Same thing. So this should be similar to the water you were using in in part 1. And then this should be lukewarm, kind of in the middle. So before you actually start doing the experiment for real, check them out. Take your finger and put it in here, put it in here, put it in here. You should be able to tell cool, warm, somewhere in between. So that's going to be important, that they're different enough that you can tell they're different, but not so extreme that they are taking you into pain on either side of cold or hot. So that's the setup. And then this is where you're going to have to just remember or dictate it to somebody who's going to write down, because both your hands are going to be wet now. You're going to take one hand and put it in the cool water, one hand and put it in the warm water, and describe what it feels like in the cool. It's you better say this hand feels cool, this hand feels warm when you first go in, or else the water is not the right temperature. But then you keep them in there for a couple of minutes. So you're going to leave them in here. What do I have here? 2 minutes. Heck. So for 2 minutes, they are staying in their respective cool or warm. And then at the end of 2 minutes, you take both of those arms and you put them both together in the lukewarm. It's worth wiggle your fingers. If you've ever been in a hot tub, you kind of know that if you're just staying still in water, you kind of get this little insulation layer. And when you move, you really feel the temperature of the water, right? If you've been in a hot tub and it's like, I'm okay, and then I'm moving around, it's like, whoa, it's hot. And it just stop moving and it's not quite as hot. It's because you kind of create this kind of insulation zone right against your skin. So if you're wiggling your fingers, you're going to make sure you're really feeling the temperature of the water. So, and again, describe the temperature sensation in the right hand and the left hand. Right? Say my left hand feels this temperature in terms of temperature. And again, not tingling or wet or this or that. It's like my left hand feels what in terms of temperature. My right hand feels what in terms of temperature. This is going to be dealing with this idea of a negative after image. Right? If things have been particularly warm and you go into the lukewarm, things that are kind of more normal temperature, if you've kind of equilibrated, adapted to the warm, things are going to feel cooler than they would have otherwise. If you have adapted, equilibrated, so cool is the new normal, and then you go into this lukewarm, this is now quite a bit warmer than your normal. So you have two hands that have adapted to different conditions. And this idea of negative after image is, what does this exact same water, both hands are going to be in the exact same water. And if you do this right, you're going to find that each hand has a very different experience. It's really kind of bizarre if you've never done it, to see how the exact same thing, again, we always think our senses kind of tell us the information about what reality is, but you're going to see that you know, it's not quite as simple as that. The exact same reality can be sensed very differently depending on, you know, this preconditioning, this pre-adaptation to these different waters. So I would really encourage you to kind of do this, set it up, you know, and really notice the temperature sensation in the right and the left while they're both in the exact same water. You know, and then, you know, describe that in your lab notebook. You're going to describe what you notice. What does the temperature experience perception of the right hand and the left hand? You know, this negative after image is kind of trippy. It works on lots of different things. They've done these studies where you show, you know, you have a picture of just some generic person here. You know, and you have to rate them on how attractive they are. Like if you show somebody a bunch of pictures of really attractive people, classically, whoa, dreamy, hot, la la la, and you show somebody else a bunch of like, ooh, they're like, you know, not, you know, they're not going to get a modeling job or whatever. And then you have them look at this face, the same face will look different based on this kind of negative after image. If you've just been looking at these kind of homely faces and then look at this average face, it's going to look a lot more attractive. If you've been looking at the supermodels and look at the average face, it's going to look more homely. So it's kind of fascinating. That kind of idea of that adaptation and then negative after image is not just temperature. It gets into these more complicated. And we're going to see in terms of your visual stuff with motion after images, you can get really cool, weird illusions. If you've adapted to things being moving and then things aren't moving, you can see all sorts of crazy, weird stuff. We'll go into that next week. And in fact, we'll do more of this. I'll show you, I'll give you a link to some cool stuff showing this kind of work as well. Like what the faces you've seen before can really affect what the faces look like afterwards. So any questions about the temperature stuff? This is something you will have to do at home. I'm going to talk just another like few minutes and then we'll give you, like, especially if you've given yourself till 12:30 to do your anatomy lab, you know, you'll still have like 15 or 20 minutes to just get out some bowls and just do it and write down some stuff. So the last part of the lab, we are not doing part 6. Part 6, you can just don't have to paste in. Forget about part 6. We're not doing part 6 at all. So maybe I'll just write that out. Not done. So ignore part 6. So the last part that we are doing, part 5, is about pain. Pain receptors, they're also called nociceptors. Pain we call nociception. Nociception is the sensation of pain. You know, before we get deeper into this, we can talk about what is the stimulus for pain, right? The stimulus for touch is stretching of your skin. The stimulus for temperature receptors is changes in temperature. You know, the stimulus for vision is light. What is the stimulus for a pain receptor? What turns on a pain receptor?
Would heat be one of them?
So heat though, but only if it's hot enough to go beyond kind of what we would think of as heat. Like I said, any kind of receptor can be perceived as pain if you take it into an extreme. So, but what happens if it's too much hot? What happens? Why don't you want things too hot?
Because it'll burn you.
Yeah, it's going to damage you. So this is about damage, or impending damage. I should say actual or impending, to your tissue, right? If your tissue is getting damaged or is in some extreme condition where you're worried that it's about to get damaged, that turns on these pain receptors, which tends to be a pretty averse kind of experience. Like you want to avoid this, you want to avoid tissue damage. What do you think about the adaptation of pain receptors? Do you think pain receptors adapt very well? No, these don't really adapt. Right? It doesn't really make sense. If you have tissue damage, impending tissue damage, it's like you don't want to let's forget about that and move on. It's like you got to deal with it. It makes pain management actually tricky. Like people who have chronic pain, it's kind of tricky to deal with because it's hard to kind of get the system to turn off. There are things we'll talk about, the natural opioids later. They basically will actually inhibit the sensory neurons from releasing their neurotransmitter in the spinal cord, so the message stops, never gets to the brain. So like if you get a shot of morphine, part of the reason that helps you deal with the pain is the pain messages are getting blocked from ever reaching the brain. But in general, pain receptors don't adapt. The other receptors we'll talk about a little later that don't adapt are ones that have to do with proprioception, like your position of your body. Like it would be pretty bad if every time you stopped moving, all of a sudden you felt like just a disembodied spirit because your whole sense of your body just faded away. So a lot of the receptors that give you basic information about your body position and stuff, those don't adapt either. Yeah, because that would be kind of freaky. Most other receptors adapt. Even vision adapts. Especially if you're in kind of low light and you just really relax and stare, everything's going to start graying out on you. It's actually kind of trippy. Typically, you don't notice it because you're always doing little saccades, even micro saccades, little twitches. You're moving, you're kind of changing what image is landing on your retina. But if you kind of paralyze the eye, everything will just start graying out if you are not changing the image landing on the retina. So it's pretty unique to not adapt. In terms of our lab, we usually use the lab as a way to introduce the idea of referred pain. Referred pain is this idea that when you have pain if you have pain on your skin, you can usually localize it pretty well. It's like we have pretty good look, like, ouch, I got a thing sticking me in the top of my shoulder, ouch. You do have pain receptors in the inside of your body for your organs, your heart, your kidneys, but they're not as good at localizing where is that pain coming from. You might something's off, but it's not like, oh, my right atrium is really experiencing a twinge right now. But what's weird is a lot of time when there is pain in your organs, it gets experienced as pain on your body. The classic, if you're having a heart attack, A lot of times people experience a heart attack, like my shoulder, my left shoulder or arm is like sore. There's pain here in my shoulder, but it's actually, there's no pain in the shoulder. The shoulder's fine. What's happening is this crosstalk between pain messages from the heart that go to the brain. And so I can explain it kind of like this. Like here's your brain and spinal cord. Ack. Oh no. Oh dag nabbit. All right, my iPad, I didn't watch the battery and my iPad just ran out. So the basic, so I'm just going to use my words. The basic idea, there is a sensory, let me turn off my squirrel so I can just show you this better. You know, if I have pain receptors in my arm and they transform tissue damage or whatever into electrical signal, and then it's an action potential, goes down an axon, goes into the dorsal root ganglion of near my spinal cord, into my spinal cord and up to my brain, to my somatosensory cortex. And when my somatosensory cortex gets a message that it knows is starting here, it says, oh, that's pain in your upper arm, shoulder. So I have the experience of pain in my upper arm and shoulder. We call it label lined coding, that there is a label, a line starts here, goes to a particular part, that homunculus in my post-central gyrus. So my brain knows there's activity here that I know started in my arm here, and my experience is pain in my arm. The pain, the neurons that carry the signals from the heart also have to eventually join up with these nerves and enter into the spinal cord and go up. And the best understanding of what's going on is that these cables kind of come together for part of the way. And what happens is there's this kind of crosstalk. So if I have these two cables coming in, and this one is, the heart is in pain, but it's actually causing some activity in this one now that's lying right next to it. So all of a sudden, the brain is getting some messages that normally would mean there's pain in my arm, even though there isn't, but it doesn't know that. All it knows is it's getting the message that says pain in arm. So my experience, my subjective, perception is there is pain in my arm, even though it's actually starting from my heart. So that's why they call it referred pain. It's pain that is in my heart, but is actually referred to another part of my body and feels like it's coming from my arm. And there's lots of different examples of referred pain, like your kidneys and your lower back or this or that. It's actually a very useful diagnostic tool, because a lot of times when people have pain and their deeper organs, it actually shows up as experiencing pain on their body. Our lab actually has a little thing where you put your elbow in ice water. So your elbow is in pain, but then this ulnar nerve, which is carrying the signals from your pinky, is getting the crosstalk of the pain from your shoulder, from your elbow. And eventually you're going to feel like your whole hand, it hurts, even though it doesn't. Your fingers are happy, but you're still going to feel like, oh, my pinky is like all cramped and cold, even though it's your elbow that's all uncomfortable. You don't have to do this at home. If you want to, you can stick your elbow in ice water and experience this, follow the procedure in the book. Or else, minimally, what I would like you to do is under doing the general sensation lab, oh, you dirty dog. Hold on. I am experiencing two-factor authorization, which drives me crazy. 1672. Hold on. 1667. Oh, where did it go? Hold on. One, 672. 361. All right. Let me in. All right, 2-point discrimination, tactile localization. Again, you can watch these to help you see how it's done. And if you don't have your study buddy, you can actually just use this as your data. Adaptation, this kind of shows you how that works. So this referred pain, watch this. Minimally, you should watch this little thing here where he's talking.
So let's talk about referred pain.
So watch that, in your lab notebook, just write a little bit describing what it is and why we care. That's the main thing I want you to walk away from this lab understanding that referred pain is a thing and that it's diagnostically important. So you should know what referred pain is. So I do want you to kind of acknowledge it in your write-up. Again, while I think I really would like you to put your hand in the water for the temperature stuff, don't worry about putting your elbow in the ice water. Normally, we pick the person in the group that's willing to, suffer for science, but, I don't want to just say everybody has to, suffer to pass the lab. And this last thing I'll just mention here, underneath, it says part 2, part 3. It says general sensation, spring to. This has the data. Did this show up on your screen? I don't have my, okay. So in your write-ups, in addition to putting in the data for what you find for your 2-point discrimination, if you do it at home with your subject, you can use this data. You definitely want to use this data as well. This data here, let's zoom in a little bit. This is saying for the face, for the back of the hand, for the palm, for the fingertips, for the lips, for the back of the neck, tactile localization, and here time to adapt with 1 coin and 3 coins. So you can use this data here as part of your write-up. Put in what you find with your particular subjects, but then obviously bring in the group data to support your discussion about what happens in parts 1 and 2. And just for a spoiler alert, you get something like this. When you look at how close can this 2 pins be and still know it's 2 instead of 1, like big surprise, fingertips and lips are good. But this usually is a big surprise. Face, you think of your face as being so sensitive. 15 millimeters, that's over a ½ an inch. You can have 2 pins over ½ inch apart on your face. You still have no idea if it's 1 pin or 2 pin. So, like I said, this lab is usually pretty fun in terms of realizing we're not as good as we think in terms of detecting what the world is around us. The same thing for tactile localization. On your upper arm, on average, people are like about a half inch, they're wrong by a half inch or forearm, when they're trying to find out where someone touched them. They can get it like within a half an inch and that's it. So this idea that, oh yeah, of course, if someone touched me, I could know where they touched me. It's like, yeah, you can get close enough, half an inch away, but that's it. With 2-point discrimination, you go to the calf. Look at this. You can have things that are 30 millimeters. That's over an inch. You can have 2 pins that are over an inch apart on the back of your calf, and you have no idea if it's 1 or 2. Right? So it's really kind of cool as you do this and get a sense of some stuff you're good at, and then some of this, and it's very uniform. Different parts of your body are going to have very different abilities to discriminate. It's ultimately going to be based on the distribution of touch receptors. So, any questions about all of this? All right, so so over the, either this afternoon or sometime over the weekend, take a little time, fill some bowls with water, record your observations. Find somebody who wants to play physiology with you and see how well they do at feeling 2 versus 1, or it's kind of funny, like at the end of doing the 2-point discrimination, sometimes it looks kind of like you've got the measles or something because you've got little dots all over yourself. So all right, so that's all I've got for the lab. And again, review the idea of like sensory transduction, because that's going to be an ongoing theme for the next couple of weeks, is this idea of transforming some stimulus, whatever form it is, into electrical signals, right? Your kitty kind of bushes your hand and you feel that kind of softness on your skin. But really what's happening is that kind of little touch on your skin is being converted into an electrical signal by the receptors going up the nerve and going up into your somatosensory cortex and your postcentral gyrus, creating electrical activity, which then creates this experience some magical way we don't quite understand of, oh, my cat's spooshing me on my hand. So anyway,